Mornings are like antioxidants, which boost our metabolism and keep us motivated to work throughout the day. So, if you also feel the same and abide by the same, you have come to the right place. Hello and welcome. This is Himanshi Kaushik, your host of the day, and I would like to welcome you all in today's webinar on oxidative stress and antioxidants, mutual player altogether. And I hope that you all are safe, sound, happy, and healthy um, at wherever you're taking this webinar from. So before we move on and learn a lot more about antioxidants in the session today, I would like to request Dr. Gurmeen ma'am to kindly introduce us to our expert here.
A very good afternoon to all of you. Today, we have a resource person, Dr. Minhaz Ahmed Khan, who is working as an associate professor and head of the laboratory, School of Bioengineering and Biosciences, Lovely Professional University. He completed his master's and PhD in biochemistry from Jamia Hamdard University. He is having more than 10 years of experience in teaching biochemistry, molecular biology, genetic engineering, and other advanced life sciences subjects, along with research in the development of biocontrol formulations against plant pathogens. His awards include UGC Research Fellowship in Sciences for Meritorious Students, which is one of the prestigious awards given by UGC for excellence in research. He was also awarded with the best poster for PhD work at an international conference at JP Institute of Technology. He is having more than 20 publications in peer-reviewed journals. He is currently supervising 5 PhD students and has supervised 25 master students till date. Moreover, he has also filed 3 patents out of his research work. I would like to thank Minhaz Ahmed Khan for choosing a topic of great relevance and correlating it with our daily lives and food. So antioxidants is a general term for any compound that can counteract unstable molecules called free radicals that damage DNA, cell membranes, and other parts of the cells. Because free radicals lack a full complement of electrons, they steal electrons from other molecules and damage those molecules in the process. Antioxidants neutralize free radicals by giving up some of their own electrons. In making this sacrifice, they act as a natural off switch for the free radicals. And this helps break a chain reaction that can affect other molecules in the cells. But it is important to recognize that the term antioxidants reflects a chemical property rather than a specific nutritional property. Free radicals are damaging by their own very nature. So therefore, they are an inescapable part of life. The body generates a lot amount of free radicals in response to environmental insults such as tobacco smoke, UV rays, and air pollution. But they are also a natural byproduct of normal processes in cells. Since free radicals are so pervasive, you need an adequate supply of antioxidants to disarm them. Now, I would like to request Dr. Minhaz Ahmed Khan to start with the session. Over to you, Minhaz Ahmed Khan.
Sir, I'm very, very sorry to interrupt. I think you're on mute, sir.
Am I audible now? A very warm evening to all of the future aspirants. And I'm really thankful to Ms. Himanshi and Dr. Gurmeen for such a nice introduction to the title today. And before I start this session, my very sincere request to all of the listeners to please forget everything right around side themselves and just be focused that what you're going to learn today. Because this title, although looks like a stressful target itself. So today we are going to deal with, first of all, that what we are as a personality dealing with daily life in the modern day life. This is the biggest challenge we are facing. And if you know this word, I think at all age groups, we all are very much familiar with this word stress. Dear friends, it is not a scientific word I'm present and at present I am telling you. This is the word being used by all communities. Every day, we are dealing with stress, which is associated with our financial burdens, socio-economic statuses, the peoples who are having their money indulged at various parts of the world. And not only that, there are a whole lot of figures around. You can see onto the picture right now in front of you that is giving you a whole lot of stress. So if you see, there is a saying by the British cardiologist, Lord Hodder, that the stress is due to work, due to lack of exercise and sleep, increasing sense of insecurity, and the anxiety connected with the competition of living. Yes, it is the right thing. The survival of the fittest. We all are struggling in our everyday life, and this makes our life stressful. So dear friends, this is the stress we are going to deal with every day. But in return, actually, what we are doing with ourselves. And if not, then today's session will open up your eyes to understand that after dealing with the stress in our daily life, what is the prevention we are doing to it? We are suffering from a number of diseases at a very younger age in the current day scenario. And the only reason behind that is stress. So what this word stress is in terms of biochemistry, we are going to understand this in today's session. Then you will be able to understand free radical and antioxidants. I'm not going to give you any more textual part of the slide, but if you can see a very nice and beautiful picture in front of you, we are all going to deal with this. I think majority of the listeners are having chemistry background, and we all know what an atom is. We all know that what an electron is, and we all know what an octet means. So any stable atom is having a paired set of electrons in their outer orbit. So what happens to that healthy atom? How we are responsible to convert the status of that healthy atom to a free radical. Friends, you can see the status of free radical in the center of this slide. It's looking like a robber who is robbing an electron from a healthy atom. So I think it gets a better picture that why it is robbing an electron, because it tries to be stable. How these free radicals are generated, that you will see in the upcoming slide. And then we are going to deal with understanding that how you can get rid of those free radicals. You can see the third hero of this slide, the hero of the third part of the slide, that is the person who is donating this electron to the one who is losing it. So this is all my session is going to cover up with the lab understanding. And my colleague, Dr. Gurmeen, has already given you complete idea on the antioxidants and free radicals. But to deal with the laboratory aspect, I have my PhD scholar, Ms. Kajal, who will be demonstrating that how you are going to understand the antioxidant potential of the natural food product. So friends, if you can see the picture in front of you, the oxidative stress, you can see there are three different parts of the same tomato. On the very right-hand side of your screen, I can say the left part of your screen, there is a healthy tomato. That what happened to that healthy tomato? That stress has converted this into a very, very rotten article. And we don't want this kind of article when we are going to purchase any vegetable in the market. But try to understand, this will be the scenario of your cell when you are getting completely attacked by the free radicals. Friends, this is what I am going to talk today. That which phenomena is responsible for the production of this kind of bad scenario. Because when we are talking about this oxidative stress, we are going to talk about this chain of bad reactions. You can see here again that there is a healthy stable molecule which is converted into an unpaired unstable molecule, because you can see there is the loss of an electron. There is an electron missing. But the problem is that this could not keep itself to its own. Instead, it will be converted into a chain of event. You can see at the below point of this, that this free radical generated will keep on asking for an electron to the neighboring atom. And that particular free radical will again ask to the next atom. And this chain or cascade of events is going to generate a huge lot of problems in our body, because we as a biological system has lots of metabolic pathways that is moving around inside. And due to this bad impact of free radical, this whole metabolic pathways are getting disrupted. Friends, try to understand why we are talking about this, because if you look at this present slide, you can't get rid of this. You can't get rid of these junk foods in the current day scenario. Tobacco, smoking. You may be not a primary smoker, but you can be a secondary smoker. The amount of pollutants you are inhaling is more than that what a smoker can inhale. Alcoholism, another such bigger issue. Air pollutants. Every now and then you are hearing or listening into the TV channels about the status or scenario of the pollutants in your country, in major metro cities of your country. Why? Because we want to be developed. We want to be more modernized. We want the much better version of the car, automobile. But we are not looking at the pollutants generated from them. We are talking about e-rickshaws, definitely. But what about the lead present in the batteries of those e-rickshaws? In one or other way, we are inhaling pollutants. And that could be a reason also for generation of free radicals. Pesticides. You are consuming pesticides in your daily food articles, through vegetables, through fruits. In your body, if they are generating the free radicals, they are going to directly interfere with your metabolic pathways. Not only this, there is another term in front of you, ROS. Suddenly, you might be thinking or wondered that what is this ROS? So friends, if you are having notebook around you, maybe you have mentioned the word oxidative stress. Maybe you have mentioned the word antioxidants. Maybe you have mentioned the word free radicals. If not, then please mention it, because there are a whole lot of questions that should arrive in your mind. And I want that you should ask a number of queries too, because this is directly associated with your life, because this is biological systems study. And in biological system, there is two things. One is life, one is death. So life is completely a part built up from the association of these biomolecules, and death is because of the dissociation of these biomolecules. And friends, this word ROS is not different from free radicals. Instead, this is very much similar to that, reactive oxygen species. If you can see inside the box of this reactive oxygen species, there are varieties of such free radical generated by the presence of oxygen. There is not much of the difference, dear friends, in between ROS and free radicals. Here you can see the major difference between both of them. But still, I could say, when I'm saying free radicals, generally any group can come in our mind. But when I'm saying ROS, reactive oxygen species, oxygen's presence is mandatory. As you can see in the examples, when we are saying free radicals, it could be a triplet of carbene, it could be hydroxy radical, it could be hydrogen radical, anything which is unpaired, which is having unpaired electrons. Similarly, when I'm saying ROS, it is peroxide, superoxide. You may have heard about the use of hydrogen peroxide in the labs. So friends, students, colleagues, whosoever is listening to this, it is my special advice to focus onto this particular part of the slide, which is giving you a very clear picture of understanding the effect of those free radicals. You know, DNA is deoxyribonucleic acid. Many of us knows this. We know that the situation inside the cell, the location of DNA is inside the nucleus. That is our genomic DNA. That is very protective structure because DNA carries the messages, the genetic information. It is that thing which we are getting from our parents. And it should be very stable molecule because this is deoxyribose. It is having an oxygen less as compared to RNA. And that is why this is very stable molecule. But friends, what we are doing by the generation of free radicals, those free radicals has the tendency to attack onto this DNA. You can see the picture inside the picture, the DNA is getting damaged. By whom? By these free radicals. You can see a whole lot of free radicals in the slide is targeting the DNA. It's not a good thing. It's a worrisome thing because in the modern day stress life, it is we who is responsible for this. Maybe you say that nothing is happening to me, but these changes are not an instant changes. They will be keep on getting changed and a time will come when you can't do anything. You can see UV light. How can you get rid of UV light from sun's rays if it is coming to you? You can't get rid of a smoker who is smoking around you. Pollutions, you can't get rid of it. Inflammations happening inside your body. The more you are getting infected, more inflammations are there. Friends, this is the way we should deal with that. Okay, if we are really concerned, then what is the solution? What should we do? Power of free radicals, you could not deny it. But how will you deal with this? Because under normal circumstances, the equilibrium scenario is that you are going to deal with all this through antioxidants. So I'm pretty sure you are very well clear of free radical oxidative stress. So all these stresses are due to the presence of these oxyradicals. Particularly, it is dealing with oxidative stress. But the word antioxidants is the word which we are going to use for the prevention. Yes, you heard it right. Note down, this is the another term which I would like to see at the end of the session. You could ask a question associated with this. So antioxidant, if you can see in my first part, under normal circumstances, it is balancing this ROS part. But under the problematic situation, under oxidative stress, you may better see who is getting more weight. It is ROS. Now you could be feeling more happier by looking at this slide. It's a very cool version of our daily life. How many of us are following this? I don't know. But when people are saying that start incorporating this in your daily life, we feel it's a joke. You can, one very basic fundamental, I could say, incorporate more of these colored natural products. These are having pigments in it. Lots of flavonoids. And these are considered to be the best antioxidants. You can see lots of Such examples are around us. Peoples of older age are much more diverted towards this because they have antioxidants. So those free radicals or reactive oxygen species are oxidants, and that is removing that electron. And these are the antioxidants that is donating an electron. So friends, this is the basic fundamental of this lecture, that the basic ingredients we want in our life is not the junk food. It's not the fast food. It is these foods that are mentioned in front of you. Very rich in antioxidants. So what will you do? Because you are here to learn from LPU, the lab of LPU, where we are going to perform a simplest experiment to find out the antioxidant power of this very, very, very demanding tea of our country, and not our country, in fact, the whole world around, that is green tea. This is the herb that is being in high demand when people are waking up in the morning. And I'm promising you, students and friends, that when you start converting your daily routine life by removing the regular tea that you are consuming with this green tea, you will see a whole lot of changes in you. I hope you may have seen so many commercials associated with this green tea. So today we are going to use this green tea in our experiment to find out, is it really having that antioxidant potential? So friends, what we all need to do in this experiment, it's a very simplest thing we can perform. What all are the requirements? If you can see here that there is some chemical requirement. Obviously, when you are going to perform in the lab, there is some minor requirements. Here you can see the one methanol is already, maybe you are very much familiar, because some of you may have done that in the lab. And some of you may have not used it. So you will see very shortly when Ms. Kajal will be showing all those things to you. There is another chemical, DPPH, that is diphenyl picryl hydrazine. That is 2,2-diphenyl picryl hydrazine. It's a complex name. That's why I'm shorting it as DPPH. Very famously available in the market. In the labs, people are easily getting this. And along with this, I'm also considering ascorbic acid as a standard. Friends, make sure that whenever you are performing any experiment, you are using a standard. Means a standard is something which is from which you are taking the reference values. Means the test sample here is the green tea, and you are going to compare it with the standard. Standard here is ascorbic acid. This is vitamin C. You may have heard about vitamin C in your corona times also, when doctors are suggesting to consume this every day. And this is the reason why. Very good antioxidant. So ascorbic acid is a standard. Green tea is the sample to find out where and how much it is having that antioxidant. So what is the role of DPPH? DPPH is the free radical we are going to use here, friends. So the antioxidant is something which is going to scavenge that electron, or to just, okay, I'll be just, sorry. Is that yeah. Am I audible? Right. So slightly this communication. So I think this is clear to all of you. Not only DPPH, Not only green tea, we also need methanol, because you want to suspend this DPPH in methanol. You are going to make the suspension of DPPH in methanol, and ascorbic acid also in methanol. So your solvent should be uniform. So this DPPH is very sensitive, very light sensitive. It is darker in color and violet in color, dark violet in color. So this we need to reconstitute in methanol. Ascorbic acid we will reconstitute in methanol, and green tea extract, that I have already got that prepared, because it takes time around 1 hour to get the extract ready. So that green tea extract is ready with us. It takes 1 hour to just use the green tea with that methanolic solution to extract out the tocopherols, that is the flavonoids present in it, to see the effect of antioxidants. So we have already prepared the extract, and then so what you are going to observe in this, here you can see the structural aspect of it. So if we want to see basically, so you can see here that DPPH, this is the free radical, and once the antioxidant is being added, you can see the free radical is now converted into a the complete ion over here. That is after the donation of an electron. So this is the principal part of our experiment, that I'm going to use an antioxidant against a free radical to show the power of antioxidant to donate the electron. So here the color change you can see from violet to yellow. So you will be observing the color change from violet to yellow. That is, the violet color is of DPPH. It is converted to yellow color in presence of green tea extract. Okay, so that's the base of this experiment. Here, my friends, you can see the machine that is being used in this, that is a spectrophotometer. So that Kajal will always show in front of you once she started this. So a spectrophotometer is used to check the absorbance of that solution, because the solution is colored. So from the reading, from the absorbance reading, we will calculate the antioxidant potential. Okay, so this is the basic pattern of our spectrophotometer, how light source is being used over there, and how we are going to note down the reading of absorbance. Okay, so here this formula that we are going to use, that simple, you can also note down this, that is the scavenging activity of my antioxidant. So here you can see AC minus AS. What is AC? AC is the absorbance of my control, right? That is the DPPH. So control is my DPPH minus AS. AS is the absorbance of my test solution. That is the DPPH plus my green tea extract. And divided by, again, the absorbance of control into 100. This formula you should remember, you should note down, right? So before I move on to the next part, I want Miss Kajal to switch on her camera of laptop, and please, we could be starting with the experimental part also. So Miss Kajal will be showing or demonstrating the various aspects of the experiment. So you can see the spectrophotometer in front of you, and first Miss Kajal will be showing one by one the various tubes that she has set. So I am requesting Miss Kajal to show one by one each tubes also that is presented, that is being prepared. So one tube will be of control, that is having, please note down once again, because this is a very informative session and you will get to know certain values at the end of this session. Okay, so please note down that the first tube that she will be showing is of blank. Number one is blank. So I'll be also writing over here. So just give me a second so that I could start writing also.
Sir, if possible, can we have Ms. Kajal on full screen, if possible?
Yeah, Kajal, can you please make it a full screen?
No, as in, sir, like we want you to stop sharing so that she comes in the full screen, because she's just visible in the corner. Is that
no, it's not required. She will show the tubes on the camera.
okay?
Yeah, Kajal, please show the blank first. So this is, friends, you can see this is blank, right? So the blank is having methanol. Right, good. Second is what we are going to show. So we are having control. So you can see, Kajal, please, yeah, more closer to the camera. Yeah, good. So this is my control having only DPPH, and you have seen control is of violet color. Good. So what is my third one? Yeah, so this is my standard. So standard is having ascorbic acid plus DPPH, right? And what is the next tube, Kajal? Yeah. So my next tube is with green tea extract. So I could say this is my test sample 1. Am I right? So this is having green tea extract along with DPPH, right, Kajal? Okay, this is only diluted sample of green tea extract. So this is having green tea extract plus methanol. And similarly, we have made certain dilutions with methanol. So let's say this is 1:100. She has also made test 2, which is having same, but ratio is 1:200. And then next is same, but the ratio is 1:500, right, Kajal? Right. So what we need to do, what we were supposed to do is to make all this earlier. And after that, you have to add DPPH in all of them, right? And if we add DPPH in it, we are adding DPPH one by one in each of them. Let me tell you, we have taken 0.5 ml of my sample here plus 3 ml of DPPH. Same is here, same is here. And we have incubated it, right, Kajal? We have incubated at room temperature in dark. Again, I am saying note down that this is very light sensitive reaction. So we have incubated it for 30 minutes. You can say 25 to 30 minutes. Now that part we have already done. So we have kept our tube at 4 p.m. and now it's already 4:30. So we have taken out the tube. And now I will request Ms. Kajal to show the cuvette to all of the listeners that how this machine will use the sample holder. So this is the cuvette. I'm writing the word cuvette. Right? So cuvette, you can see here, it's a cylindrical structure, something like this. And the capacity of this cuvette is 3 ml. So one by one, Ms. Kajal will start taking the reading in a spectrophotometer. Right? So first she will be taking up the blank, and by that time she will be taking the readings. I will be mentioning these readings from the spectrophotometer in front of you. So Gurmeen ma'am will be assisting Ms. Kajal also to show the reading display in its spectrophotometer. So Kajal, first you keep it onto the blank. Yeah, so this is my spectrophotometer and this is the sample holder and the blank is being kept. Make sure that the reading of blank will be 0 and there should be no absorbance, right? So my blank should always coming up with the value as 0. And then we will be using the control. So I would like to write the value. So first my value is 0.000, and then second is for control. Can I get to know the reading, Ms. Kajal? So my process of DPPH reading is under process going on. So I'll be just writing the words, the numbers very soon. And then she will be taking up for ascorbic acid with DPPH and followed by all the test samples. So what is the reading I have got? 1.4. Right. So I would like to see for, as per the formula, if you see, right. So this is for standard 0.015. The reading that I have got is 0.015. And now I will be writing for all these three samples one by one. That is, these are three my diluted ones. So as per the formula, the standard, that is standard minus control divided by standard into 100. So this will be showing the percentage scavenging activity. This is for, all right. That is in process. So we have in front of you the readings of ascorbic acid standard minus my sample. Point. All right, so I'll be changing up these. Let me remove, let me erase this. All right. So, yes, thank you, Gurmeet ma'am. I'll be taking my. Right, so let's say the standard I have got over here is, yeah, 0.195, and the second one is 0.085 divided by 0.195 into 100. So we will be calculating this, that is 0.195 minus 0.085 divided by 0.195, right? So let's say 0.195 is that of standard minus 0.085 divided by 0.195 multiplied by 100. So what is the answer? So I hope many of the listeners could have calculated the answer also for this particular box. Maybe you are excited also to write down that in the chat box. So this is the values of my standard, my test sample, and my standard again, because I have told you this is the formula, multiplied by 100. So as per my calculation, this is coming out as 56 point something. So the value is saying that this is 56% of scavenged. So friends, this is, I mean, that's something very, very, crucial at this stage, that how much your green tea extract has shown its antioxidant potential, that is 56% in scavenging that free radical, which is DPPH. So I hope you may have understand these facts, these understandings of free radicals, antioxidant, and once again, students, just in a faster recap, I would like to tell you that the utilization of antioxidants, one minor example I have just shown in front of you by looking at these calculations, that just using a green tea is showing you that in this time span of 30 minutes, it shows the scavenging potential as 56%. This is the power of antioxidant. And why I'm telling you all, because every now and then in modern day life, you may be listening that someone is dying of cardiovascular disease, someone is having cardiac arrest at the age of 24, someone is dying because of cancer, and today there is no age limit for any disease. And friends, that there was a time when people says chalta hai, hame kuch nahi hota. Nowadays you can't say these things, because ab sab kuch hota hai. In the smaller, simpler words, I could say, because you can't get away from these pollutants, from heavy metal pollutions, from the pollutions generated by our own house. How could you get rid of the sun's rays? How could you get rid of UV rays, which is having the sun rays is coming directly? You may have heard that UV rays is striking you directly. The lead used in the industry, the effluents coming out from our own houses. We can't get rid of these pollutants. So obviously they will be generating free radicals. What is the role of free radical? They will be creating the cascade of events to generate more of free radicals. You can't stop. Even antioxidants is not promising you that I am stopping the free radical generation. No, don't make a bad concept. Antioxidant says that I will try to convert these free radical into a stable atom. That's the concept. So that is why what you can do, you can try to incorporate these foods, which I have shown during my presentation, that incorporate this. You will be, getting rid of all those, what I can say, the bad activities of free radicals. And I am pretty sure that with all this, maybe you may have enjoyed the session. You may have learned about free radicals and antioxidant. So if you have any questions, any queries, you are most welcome. Over to you now.
Okay, sir, thank you so much. First of all, before we go to the was so good, and practical point of view is again a cherry on the top. It's like we know, okay, these things are done like these, and all the information is really, really, really appreciated, sir. Thank you so much for coming in, for joining in, for crediting your time and experience to us, to all the participants. Thank you. And thank you so much, Gurmeen ma'am, also for coordinating so well and for coming in and for joining in. And thanks to Ms. Kajal also for showing us the experiment, the main part of the session. And sir, with your request, we have few queries from attendees. So can I take them?
Yeah, I would be happy to answer those queries.
Okay, okay. Sir, am I audible properly, right? I'm audible
Some
properly?
interruption was there, but it's okay. We can manage.
Okay, okay. So this first, okay, so anonymous attendee has asked this question. Sir, then how can we save ourselves from pollutants?
As I said, it's really difficult, really difficult. You know, you may have seen the ad of Fair and Lovely cream, right? So when the girl is going around in the sun and suddenly the heroine is saying ki, just use this Fair and Lovely cream, you will be, so fine. But actually these all are a part of commercials, but basically you can't get rid of that. Uh, it's all about the UV rays. It's harmful UV rays. It has a tendency to penetrate in the DNA. The question obviously will be arriving in the mind of these younger generation that how to get rid of that. I'm saying it's very difficult, right? Although you are covering up whole bodies, but do you think UV rays can protect it? No, it can't protect UV rays. So the main fundamental is when you are moving outside, you are inhaling lots of pollutants. You can't stop those cars. You can't stop those automobiles, right? So industries are generating whole lot of, we are very well versed of sitting in ACs. People are using generators even to run the ACs, but in generators we are using diesel oils. So the pollutant, you can't stop them, right? So the only thing is consume more of antioxidants. Start consuming more of antioxidants. Again, I would say antioxidants can't stop it, the generation of free radicals. The important thing is it will at least try to replenish the repair damage part could be done by antioxidants.
Thank you so much, sir, for resolving this query. And we have another query from HKK. So this query reads, is there any way we can stop free radicals production?
No
It's kind
ways.
of
No ways. Basically, free radical generation is something that's not happening in front of you. It's a part of our metabolic pathways. It's going on around. So, the only thing is ki, as people are saying ki it is being written smoking free zone or pollutant free zone, and this area is very much purified air is there. So try to avoid those polluted areas, polluted places, because free radical generation, it's not only dependent upon pollution, instead our fooding habits also. As I said, people are moving towards the junk foods more, they are more moving towards the working foods more. So generation of free radical is very much always in process. So if you are taking fried foods, junk foods, it is better to have more of these incorporations of antioxidants too in your diet.
Yeah, I totally agree, sir. Sir, there's one personal question from my end. We have seen commercials of air purifiers. So, I mean, do they really purify the air, or is it just for the commercial use?
If we try to categorize the pollutants on the basis of the size, you may have seen that even certain commercials are showing that the water purifier are getting rid of viruses, bacteria, but it is of very 10 seconds commercial. They can't categorize the size of the microbes or the pollutant. So depends upon the size of the particle. So if you see the pollutants in around here, the peoples who are claiming that the air is purified, or even these air conditioned peoples are saying our air is more purified, but technically the air could purified from what? That's more important thing. Purified from what? Yes, the larger particles, obviously it will stuck in your filter and it won't come in. But what about the smaller particles? So there is a particular size also, which every purifier could not get rid of.
Definitely, Sam. Thank you, sir. That was my personal query. And so with this, we have another query. Okay, this is something that has happened. So it says, sir, if you can tell me what happened to New York City, all the sky turned red. How?
Even I have seen that news also. Yes, it is because of the fire in the jungle. Maybe student has not heard the whole news. It's a fire in the jungle, and yes, whenever there is a fire in the jungle, it's very difficult to control. And yes, the government of that particular, whole government management over there, they have asked the peoples to stay back into their home. Because yes, this is something which I was saying. When people are saying I'm feeling asthmatic, I'm feeling chest problems or anything like this, even these days younger people are getting this asthmatic issues. This is of stocking of those particulate size in the respiratory channels. That's the issue. Yeah.
Yeah, sir, and we are continuously hearing news of demise of people through cardiac arrest, heart attack, very small age. So I think we need to focus more on our health in terms of the food that we eat and in terms of the environment that we live in.
Even I
Sir,
can give a very small example also, like people are using these colored paints very much liberally. It starts from the childhood days. Our kids in the home, they are taking the things in their mouth, the colored articles in their mouth. These paints contains lead. This is a direct consumption, but we are very happy when our kids play with the toys. If you go to the school, they are just licking up those swings around, and they are having this whole lot of lead pollution inside them. But if you go to the people who are working in the lead industry, you will see their crippled legs, their muted systems. Their kids are not born properly. They are the real time examples of this hazards. You are consuming it in micro nanograms, but they are consuming it in milligrams, grams. And that is the reason their kids, their birth are not up to the mark. So it begins from the childhood. Yeah.
Yes, sir, I definitely agree. The consequence of this is so much hazardous for us. We need to sincerely work on this. And so this next question is very interesting. So it says, why sustainable development does not include planting trees, but using other things that might hurt the nature?
Last part of it, I didn't hear.
Yeah, but using other things that might hurt the nature, but we do not talk about planting the trees. We talk about every other thing but planting the trees.
No. There is actually, I think this student may have not gone through all the SDGs very clearly. If you go to the SDG 15 and SDG 14, there is SDG 15 on life on land, and SDG 14 is life underwater. So yes, it includes the plantings. It includes, and our school in LPU, like School of Bioengineering and Biosciences, is very much dealing with this. So even my school head, Dr. Neetaraj Sharma, is working on the project of water treatments and at the same time the green economies. So green economy is the European project that is comprising of planting these trees, and that is totally based on SDG 15.
Yeah, okay. So I think you need, I do not know the name, but I think you need to, but okay, you have asked the question, and thank you, sir, for resolving this query. And sir, we have another question. It is, how antioxidants help us to reduce the darkening and aging?
Every time, either it is associated with aging or darkening, as I show in one of my slide of tomato where it was getting peeled off and badly rotten, that is something associated with us also. Yes, darkening or aging. This is the meaning of oxidant, is oxidation, the lots of electron. And suddenly someone comes and says, okay, take this electron, fulfill your octet. You are true. So it's not like as it is being shown in the films, ki aaj kale hue, then kal hum gore ho gaye. Like, it is not like that. It's a time taken process. But yes, darkening, aging, these are the natural phenomena. But if you keep on taking vitamin E, which is one of the very best antioxidant, it is present in orange or citrus fruits. That's what I said. Ki incorporate more of pigmented fruits, more of pigmented vegetables, and they will show you the change. So these dark pigmentation or aging process, you will see the relief from that.
Definitely, sir. Thank you, sir. With this, we have last two queries, and this query is again from anonymous.
I'm so happy people are so much indulged and they're asking queries. I'm really happy.
Sir, we have, okay, anonymous attendee again wants to ask, what can be good research topic in antioxidants for PhD?
I'm so happy and glad to hear this query that someone has been asking this. And truly speaking, my PhD students are also working onto this. That is the anti-cancerous potential of herbs. So let's take an example of the herbs present in our own kitchen also, like spices and herbs. I could categorize it like that. Like cinnamon is being used. My another student is working on some another herb, like giloy. Someone is working on this datura, and someone is working on this Miss Kajal. She's also working on to the same anti-cancerous potential. She's formulating it from one of the herb. I'm forgetting the name. So all of my students are working on to these aspects of anti-cancerous potential present in these herbs. So I'm just trying to give it as an example that even whatever spices or herbs like curcumin, like cinnamon, like turmeric, like cumin seeds, all these are having certain potential like antioxidant ability that shows the power to prevent the generation of free radicals. And that is one such aspect. It can be anti-inflammatory also, and that is why it could be anti-cancerous. So this could be the research topic. So ultimately the student may go for developing a formulation in anti-cancerous role by working on this antioxidant area.
Definitely. And to all the participants, I would like to say that as there are many students that who are doing their PhD under sir, so you can just join in here. I mean, this is going to be a good option for you because sir has already the experience of teaching students. So I think that you can just explore this option also. And sir, with this we have our last question, and it's again very interesting. Sir, aren't this green tea packet has microplastic in it?
Green tea packet.
Yeah.
Absolutely, absolutely. There are a whole lot of industries. Please do not rely much onto these pouches. So, obviously the packaging part may definitely give you. This is a very good question. Microplastics are everywhere. So if you can get the real leaves, I'm not saying the processed leaves. There is a difference between both of them. So green tea, you may get, these are the dust, which are mostly available in the pouches. And the real green tea also, you may get some very crude extract, you can say the crude leaves of this green tea. That is also very effective. So yes, the pouch manufacturing may have certain microplastics in it, but the good effects of green tea actually surpassing those.
Yes. Thank you, sir. I think we're done with the questions. Now I would like Gurmeen ma'am, if she wants to add anything to the webinar and to the topic, Gurmeen ma'am.
Yes, ma'am. I would like to thank Dr. Minhas Ahmed Khan for such an enlightening talk on antioxidants and oxidative stress. And the way he has narrated speaks volume about the expertise he is having in his area. And taking the example of green tea, which we all know as one of the biggest asset for antioxidants, has taken this whole session to an altogether different level. Thank you so much, sir, for shedding light on the importance of diets rich in antioxidants to be taken in our daily routine to tackle these free radicals. Thank you so much, sir.
So thank you, ma'am. And sir, over to you. Would you want to say anything at the end of the session?
Only would like to say that please do something, do research in the area which could help the mankind, and societal benefits are always there. So just pick up those societal issues and develop a solution towards this, because as you said, SDGs are, the peoples are working on this, and by 2050 who knows what would happen to our natural resources. So do something in this area that you will definitely get in return. Thank you so much.
Thank you, sir. Thank you, ma'am. And thank you, Ms. Kajal, also for joining in. And sir, lastly, I would take your permission to show our participants a bit about the site and the admission, how they can take admission in the course. Can I?
Yes, ma'am.
Please,
Okay,
carry.
okay. Okay. So now I would be sharing my screen to all the participants here. Okay. So what you need to do, participants, you just simply need to write this site, that is lpu.in. And once you write this site, so this will be the interface that will be coming in front of you. This is something like these are all the highlights of the university that might have happened or that might happen in the near future. Basically, all the highlights. And I would request you to kindly explore also, because I promise that you won't regret it. Now, since we're catering about admissions here clearly, so once we click on the admissions, we have all the programs that are there. And I'm going to take an example of, let's say you are a student who has done your 12th grade and wants to take an undergrad program. So you go to simply after 12th and undergraduate and then regular programs. And once you click on the regular programs, there are a whole lot of disciplines. You name any discipline, we have it here. You can see all the disciplines we have. Design, fashion design, multimedia, sociology, geography, Punjabi, etc. Now, since we were talking a lot about plants today and antioxidants, so why not take an example of botany? So once I clicked on botany, there will be the program that is offered under that. And once I click on that program, that will give you all the knowledge of the program. So these all the highlighted options are there, right? You will be getting all the knowledge from these all highlighted program options. So in just 1 minute, I will be briefing them all. So program details would give you your eligibility, whether you're eligible to take a particular course or not, to take admission in that particular course or not, you would be finding from program details. Now, moving on to highlights would be giving you all the placements, research, student achievement, and rankings of the university that is there under that particular discipline or program. Now is very important and interesting to note is the curriculum part. So curriculum basically tells you what are topics that you're going to be taught in the next 3-4 years of your curriculum of your course. And is the fee structure, I'm going to tell about the fee structure here. So fee is 50,000 per semester, but again we have other scholarships and you can avail up to 28% of program fee. So how do you check that what is your fees from here? And after that, I will be telling you an important date, that is 15th of June. And how you can apply is from here. So there you can apply the requirements, you just submit them and you will be given your dashboard. And from there, you can apply for the course. I hope that I have given you all the insight as to what you need to do in order to take part in a particular course. And with this, I would want to take your leave, all the attendees. Thank you so much for joining in. Until next webinar, stay safe and stay healthy. And I've already shared a link in the chat box, wherein if you would want to have a session with our counselors, you can simply click on the link. Thank you so much for joining in. Meet you soon in the next webinar.